We start a talk, just wanted to let everybody know that if you are not already subscribed to our mailing list, you should consider it so that you can get added to our invitation list for in-person events and get notifications about careers, samples of free content, all sorts of good stuff. There are over 6,000 drug hunters worldwide on our mailing list now, including scientists from every major pharma company and biotech company worldwide. From the US to the EU to Korea to Japan, life science VCs, people working in related areas, all find this useful. You can join by going to drughunters.com/join or scanning the QR code on the top right and entering your email there. All right, so last time we talked about 5 drug approvals from the last 6 months and 5 highlights from molecules of the month. This time, we're going to talk about everything else. So interesting applications of new technology in drug discovery, billion-dollar molecules, and a little bit of a focus on AI from our recent series. And finally, Lauren and David, who are here today, are going to share their favorites from recent things that they've been looking at. So first, we'll start with some interesting highlights of companies working in new modalities. The first, an area that I'm really personally excited about is DubTax. So everybody's heard of PROTACs at this point, molecules that recruit E3 ligases to take target proteins to the proteasome for degradation. DubTax do essentially the opposite. They recruit deubiquitinating enzymes that remove ubiquitins from proteins to prevent them from being labeled for degradation and stabilize them for various applications. So rather than degrading proteins, you could potentially increase certain protein levels. And you can see how this could be useful for elevating levels of tumor suppressor proteins, for example, or proteins that need higher levels in certain diseases, like the chloride channel in cystic fibrosis. So the original Nomura and Novartis publication on the topic used cysteine-reactive covalent ligands to recruit a DUB enzyme called OTUB1 to stabilize CTFR, and we won as a proof of concept. And it looks like they are expanding this concept to other areas with a new company. So Vicinitas, a San Francisco-based company, just raised a $65 million Series A round, led by Andreessen Horowitz and Deerfield. So very cool. Second area that's been super hot recently has been the application of psychedelic drugs to the treatment of mental health issues like depression. So Delix Pharmaceuticals is a Boston company that recently raised $70 million, led by Artis Ventures and RA Capital, to develop psychoplastogens, or non-hallucinogenic analogs of psychedelics. So there's been a lot of research showing that drugs like LSD and DMT can help with symptoms of depression by inducing plasticity in neurons and all sorts of interesting mechanisms. This has been a super hot area. I know if you're in Oregon, you can see psilocybin clinics getting started all over the place. And from talking to people who have actually tried these approaches, it does seem to actually work. Now, obviously, it'd be nice if instead of giving everybody LSD, we could give them a form that didn't have all the drawbacks, like the hallucinations and things like that. But there hasn't been a great way to screen for hallucinogenic potential. So one thing that this company is likely applying is using their co-founders' psych-like approach, in which they've genetically engineered mice to have GFP-labeled serotonin receptors so that they can see in vivo what conformation the receptor is adopting after being given drugs. And so they've used that to screen a variety of psychedelics to look for things that are specifically non-hallucinogenic, but still induce a conformation that's relevant, and have appear to have found non-hallucinogenic psychedelics. So they're taking these into clinical trials, and we'll be very interested to see how they work. A third area in a slightly different space that's interesting is finding new approaches for stable antibody-drug conjugates. So if you've been following antibody-drug conjugates, you know that one of the key issues is bystander toxicity of the cytotoxic payloads that are on the ADCs. So they do kill tumor cells, but unfortunately, one hypothesis is that because the linkers may not always be stable, some of the cytotoxic drugs can be released. They may cause toxicities to other tissues that are undesirable, leading to a smaller therapeutic index than originally desired. So typically, these ADCs are formed through potentially reversible linkages or otherwise more easily degradable linkages, such as through conjugate addition to a malayamide. Tubulis, a German company, has raised $65 million in their Series B, led by Endera, to develop some new approaches to make more stable ADCs. One of these approaches I thought was chemically interesting is they use conjugate additions to these phosphorus-containing alkyne compounds, resulting in ethynyl phosphanamidates that apparently are, ethynyl phosphanamidates that are apparently more stable. They're also using another technology called TUBTAG that I won't go into too much detail about, but they're trying a couple approaches to make more stable conjugates. And their hope is that these will have better therapeutic indexes going forward. If you thought these sorts of things were interesting, we have 14 other companies and highlights in this recent technology review article, and you can check out more of those there. We can't talk about new technology without talking about AI and machine learning. So I was really surprised to see in one of our recent webinars when I polled the audience that people were most excited about AI and machine learning as a new technology, even relative to other areas like protein degradation and other areas of induced pharmacology, proximity-based pharmacology. So why is AI so interesting? Well, I'll just start with a very quick introduction for those who are not super familiar with the field. So we're all aware of how you handle small data, right? So if you have many points of data, you can generate a scatter plot, and if you want to predict what's going to come next in your data set, you would do some kind of linear regression or some kind of plotting, get a trend line, and extrapolate based on that trend line. So big data basically just does this, whereas the scatter plot and trend line are in 2 dimensions. Nowadays, with a lot of machine learning approaches, you can predict, future data based on n dimensions of data points, right? So you can take massive amounts of data, like billions of cat videos, right? And based on user preferences or data about user behavior, you can extrapolate what somebody might want to watch next. So obviously with these abilities to do this based on, for example, voice recognition, image recognition, video processing, all this sort of stuff, the natural question is, well, in the drug discovery industry, there are so many sources of complex data. Can you apply these same sorts of approaches to predicting relevant properties in drug discovery? So you can think about any source of big data that we have in drug discovery, from properties of large sets of molecules, to literature, to genomic data, to cellular imaging data, to databases of protein structures. And from that, you could imagine extrapolating, what future good drugs might look like, biomarkers, or what good targets might look like. This has been an area of intense research for over 10 years now. So Exscientia was one of the first companies to get funded in this space in 2012. And there have been so many, over 200 startups working in this area. We are just starting to see the clinical candidates from these companies be disclosed. And one of the most notable was this SHP2 inhibitor from Relay Therapeutics. So this molecule is often cited as a good example of AI ML application to drug discovery, although they haven't disclosed much technically about how it was applied. The molecule's just entered phase 1 and has been licensed by Genentech with a $75 million upfront payment. While the compound is structurally related to the Novartis starting point, TNO155, it does have some interesting structural differences. So if you try to map this molecule directly onto it, there are areas of the molecule that don't exactly line up. So it will be interesting to see how machine learning helped them make these decisions as they start to talk more about their science in the future. Relay is also working on other hot targets in oncology, like FGFR2, PI3K alpha, And these are programs that have been challenged by selectivity in traditional programs. And so it'll be interesting to watch how, as they disclose more of their science, how their models helped them find those compounds. Other companies are less focused on novel chemical matter and more focused on novel biology. Recursion Medicines was a company I thought was particularly interesting in their approach. So they have a cell painting technique in which they use multiple fluorescent dye labels and detection channels to characterize cells with greater detail. So if you've sat in on an immunology meeting or an oncology meeting, you know that the traditional way biologists characterize cells in a well is by staining with antibodies. So immune cells, for example, can be characterized as CD4 positive or CD8 positive or things like that. But as biology has evolved, we now know that even within certain types of cells, like T cells, there can be many different states of the cells. They can be exhausted, they can be activated. And in partnership with other cells, they can have many different properties. And so we can't think about cells as just being, in little buckets, nice little buckets anymore. So what they are able to do now with, the assistance of AI and machine learning and, all this better image processing power is they can use many, many different channels to label cells and get much greater detail and insight into what these cells are actually doing, in vitro or in vivo, and how drugs are influencing these different cell states. And because of that, they're able to take drugs targeting different targets and explore their applications in new settings. So the molecules that they're pursuing are against, relatively older targets, HDAC, MEK, PKC, but they're hoping that this approach will let them find new applications of these in different diseases like neurofibromatosis for this HDAC inhibitor. Now, if you watch the news or read, any other non-scientific channel, you'll often see that people are saying, oh, the average AI/ML program is faster and there are many clinical candidates. But if you actually dig into the molecules, as we've done in our recent 3-part series, you'll see that most of these clinical candidates are actually repurposed programs. So there are many more companies trying to study new applications of old drugs with new biology than there are trying to apply AI/ML to actually improving medicinal chemistry or discovering novel compounds like Postera or in silico. So while this is still interesting, I think this just shows that applications to, finding really novel molecules for novel targets is still maturing. And that makes sense, right? There's so much data involved. I think companies really want to focus on getting good at applying AI and ML to one type of data at a time. And hopefully in the next 10 years, as we watch this mature, we'll see more combinations of not only applying AI ML to, drug properties or biology or anything like that, but combining all of those facets. And we may see that more in our daily work. So I don't know if any of you have played around with these tools before. I think these are so cool. If you go to places like deepai.org or things like that, you can enter a prompt and the AI will generate an image. Just for fun, I threw in molecules of the month and Drug Hunter, and this is what it spat out. It's interesting in a, in an artistic sense. I can see how they got this picture on the left from Molecules of the Month, right? It's a nice little grid with a couple of the letters at the top. And while this is fascinating, I think this does show, if it's this hard to get image processing perfect, I can only imagine how hard it is to apply this to making new molecules. But I'm very excited to see where the field takes us, and very impressed by the people trying to make this happen. All right, so the final section that I've got is billion-dollar molecules. It's always nice to see success stories in drug discovery where value is being created for patients and that's being recognized in development. So one of the coolest molecules, I think, recently is voxelotor. This was acquired from Global Blood Therapeutics by Pfizer in a $5 billion buyout. It is a first-in-class oral reversible covalent aldehyde that prevents sickle hemoglobin from polymerizing and driving sickle cell disease. So this is particularly notable because the aldehyde does make this a reversible covalent drug. It targets a valine residue, an N-terminal valine. And this is just, really interesting because, again, aldehydes, I think, rarely are considered good drug motifs, but this molecule is dosed over a gram daily and seems to be very well tolerated. It binds through a reversible linkage, again, to an amine on this hemoglobin tetramer. And the way it works is it stabilizes these sickle hemoglobin tetramers to prevent them from polymerizing and forming clots and leading to these really painful attacks. You can see the binding mode here. So what was interesting in this discovery story that we talk more about in this article at the bottom is they originally started with hits that bound in a 2-to-4 fashion. So hemoglobin is a tetramer of 4 subunits, and the original leads had a 2-to-4 stoichiometry where 2 of these molecules would bind to each tetramer. As they optimized the molecule, they discovered molecules like this, which actually bind in a 1-to-4 stoichiometry, where only 1 unit of the drug is needed to glue all 4 pieces of the tetramer together. In the structure, you can see the heme in the background, which I think is really cool. And so, this is a really cool example of a crystal structure of a molecule with a shift base being formed to its target. The next molecule, which is actually from an unpublished article from our team, which hopefully should be coming out soon, repotrectinib. This is a ROS1 TRK and ALK kinase inhibitor, and it has a really interesting small macrocyclic structure, kind of like an amide-containing cyclophane or something like that. Because of its small structure, it is oral and more readily CNS penetrant. And this is important for cancers because the way a lot of lung cancers progress is through brain penetration, is through brain metastases. So a lot of non-CNS penetrating cancer drugs, they'll work, but then eventually the cancer will escape to the brain, and that's not great. So ROS1 is a pretty prevalent mutation in lung cancer patients, and NTRK is a pretty prevalent mutation in these rare neuroendocrine tumors, both of which did not have great treatment options before these newer kinase inhibitors. What I thought was interesting about this approach was because they have a smaller binding footprint, and the thesis was that there would be fewer resistance mechanisms if there were fewer dependencies of the molecule, binding to different areas of the target proteins. And this appears to play out both in vitro and in the clinic, where they see longer survival, longer response durations, because they're able to escape a lot of the mutations that other inhibitors like larotrectinib and entrectinib seem to be susceptible to. So BMS recently purchased this molecule and the rest of the Turning Point pipeline for $4 billion. And Turning Point has a number of other interesting macrocyclic compounds that I think are also worth watching. The last billion-dollar molecule I'll highlight is avacopan. This is from a San Francisco company, Chemocentrics, which has been active for almost 20 years with a focus on finding molecules that modulate cell trafficking, or the migration of cells from different areas of tissues to others. Amgen paid almost $4 billion for this, and this is a first-in-class drug that selectively blocks the effect of a complement protein, C5a, through its GPCR receptor, C5AR. This results in preventing neutrophil chemoattraction and activation, and in this disease, AAV, neutrophils are pathogenic where they're hyperactivated and they go to tissues and cause, irritation, inflammation, and a lot of undesirable, painful effects. So this is also interesting because as a small molecule, it is the first approved small molecule complement pathway modulator. So the complement pathway is an innate immune system pathway that has been actively pursued for 2 decades. Ever since it was discovered, people have been trying to drug it. Most of the drugs that are used are biologics. And so this is the first small molecule that's been able to demonstrate meaningful efficacy in an indication targeting this pathway. For chemists, I thought you'd appreciate this molecule because it has two anilines or anilid or an aniline structures here. It has 5 rings and 2 amides. It's a good example of what is considered a acceptable drug is evolving. I think the bottom line is that it works in this rare condition, and is very interesting to see that they were able to make an oral molecule with 5 rings. Okay, so with that, I will turn it over to David first for his favorite molecule.
Thank you, Dennis. I have chosen a covalent BTK inhibitor. This is one of the molecules that we'll be discussing in our upcoming October Molecule of the Month report. One thing that drew my attention to this molecule was the presence of the atrope isomer located on the top left part of this molecule. It drew my attention to it because if you've been following along some of our series, we've recently put out an article sponsored by Sino Compound on synthetic access to stable atrope isomers in medicinal chemistry. These types of compounds aren't seen very often they're not seen very often in medicinal chemistry because of the fragile nature of some of these atrope isomers. But I think as the technology advances to isolate them in optical purity, that we'll see more and more of these. The molecule itself is a covalent inhibitor. It reacts with cysteine 481 in the kinase domain of BTK. It has, if you're aware of BTK, it's essential in B cell activation as well as for macrophage activation via the Fc gamma receptor. The molecule itself is fairly selective. The only other kinases that it hits are those that have a cysteine in the same location, and they're all within the same family that BTK is in. 3 covalent BTK inhibitors have been approved already, so you might ask, why do we need another one? But the problem is that many patients with hematological malignancies don't achieve a complete response, or they undergo regression shortly after treatment. And so the need is obviously there to develop stronger and longer lasting BTK inhibitors. This molecule is orally bioavailable. J&J had originally published a compound similar to this, but it had very poor bioavailability. And so their optimization efforts focused on both bioavailability and having solubility as well. The other compound had to be dosed in cyclodextrins, was really not very patient friendly. The compound itself has in vivo efficacy in preclinical. Its efficacy in diverse large cell B cell lymphomas, or DLBCL, as both a monotherapy and in combination with the BCL2 inhibitor venetoclax, which is somewhat unique to other BTK inhibitors. And as of now, it's a clinical candidate for phase 1 in hematological cancers. So look for this and 9 other compounds in the next couple of weeks when we release our October Molecules of the Month.
Thanks, David. I will pass it over to Lauren next.
Thank you. So I chose to also preview a molecule from an upcoming article. This one will be in our next Moving Targets article. And I chose an oral gamma secretase inhibitor, which is being implicated in desmoid tumors and multiple myeloma. So I chose this as a moving target because it has a recent clinical update for desmoid tumors. So it recently met its primary endpoint in a phase 3 trial. So you can read about that in the update. And SpringWorks which is the developer, is going to submit a full NDA filing package to the FDA by the end of the year. And the reason why I thought this drug was interesting is because gamma secretases in general were originally developed for Alzheimer's disease, but they've been recently repurposed for anti-cancer agents. And this one in particular, so the reason why it's being used in desmoid tumors is there's a pathway of Notch receptor cleavage, which inhibits desmoid tumor growth, but not regular tumor growth. And then there is another pathway of direct cleavage of membrane-bound BCMA cleavage. And so it's also being tested in several phase 1 and 2 trials with BCMA-directed therapies for multiple myeloma. So all of these updates will be in the next Moving Targets article if you're interested.
Very cool. Yeah, I'm sure a lot of people in the audience have probably seen or worked on gamma secretase inhibitors for Alzheimer's. So it's very cool to see this target being pursued for cancer instead. All right, so before we go on to Q&A, I'll just highlight, if you thought the molecules that we covered today were interesting, or the companies or anything like that, you can get more with our premium subscription. This is how we support all of the free content that we provide on our site and free webinars like this. We cover why the molecules matter, how the target was selected, why these things are interesting, how the hits were discovered, all kinds of interesting scientific information so that you don't have to spend 20 hours on PubMed figuring out all this stuff for yourself. Everything on Drug Hunter is made by scientists for scientists. So if you read the news, you know that you'll get quick updates and you'll see what's breaking, but it won't be put in context or science heavy because their goal is to just publish things as quickly as possible. On the other end, if you look at databases or journal articles, they're comprehensive, but they're really time consuming to read and search through because their objective is to cover everything under the sun. So what we try to do is balance this, where we try to tell you about science that's recent and interesting, but still give it to you at a depth that is useful for somebody who's got a PhD and 10 to 20 years of industry experience. So David and Lauren are two of our full-time team members. We also work with 20 other PhDs and PharmDs on all of our content. We have biopharma reviewers working at a variety of companies worldwide, and the team goes through over 10,000 abstracts a month, spending over 1,000 team hours putting together all of these reports for you. So if you would like more ideas, new insights to help you on your programs, or just discover more in general, you can join all of our corporate subscribers by asking your company for a subscription. We work with large pharma companies, VCs, startups, and academia, from Boehringer Ingelheim to Harvard to Google Ventures. So you can sign up for a trial at drughunter.com. All right, with that, we can go on to questions. Okay, so.
We have a couple in the Q&A there, Dennis, if you want to look at those.
Okay, so a question about gram-negative bacteria. David and Lauren, I feel like we covered a pretty interesting gram-negative targeting molecule in a recent Molecules of the Month. Can't remember exactly which one it was.
I believe that was 2 months ago, if I'm not mistaken.
Yeah, it was a pretty cool new mechanism. Was it from Novartis? Novartis Emeryville? Yeah, well, I'll just say if you search gram-negative on Drug Hunter, you'll probably find a couple interesting things. All right. Do we have any other questions?
There's
a
another
question about novel antibiotics effective against MDR, multi-drug resistant pathogens. We do try to cover those. Whenever I see something like that show up in the literature, I give it certainly a thorough read to see if it might be of interest to our readers. And oftentimes it will show up in Molecules of the Month if it's got substantial activity, because I think, as we all can appreciate, the rise of antibiotic resistance is real. and it's going to be a real threat. And so it's something that traditionally large pharma hasn't done much with over the past 10 to 20 years, but I think we're starting to see a light come on, and more people, more and more companies taking this seriously.
Okay, great. Well, if there are no more questions, thank you all for dialing in.